Hello, everyone. Thank you so much for joining us for today's coffee chat. My name is Cameron Lilly, and I'm a Scientific Affairs Liaison with Menarini Silicon Biosystems. And the story I'd like to tell you today is about our attempt to meet an unmet need in multiple myeloma. And that is the need for a non-invasive liquid biopsy test to monitor and screen in this disease. To do this today, I want to talk a little bit about multiple myeloma, the disease itself, and a little on how it's diagnosed to underscore the need for a new test like the one we've developed, which is the CellSearch Circulating Multiple Myeloma Cell, or CMMC, test. I'll talk about the assay's design and the cell rescue tubes, which are a pivotal part of sample collection for the CMMC test. I'll then move on to present some data on monitoring multiple myeloma with the CellSearch assay, and of course, talk about how you can bring this assay into your own studies by partnering with our global lab services. And then, of course, we'll take questions at the end. So multiple myeloma is a clonal cancer of antibody-secreting B cells, which we call plasma cells. This particular cancer invades the bone marrow and overwhelms it, and it disrupts the normal bone marrow environment, leading to what we call myeloma-defining events. This can be anemia, bone lesions, which can be localized pockets of disrupted bone, like you see in the image on the right, or even large compound fractures in long bones. It can lead to secondary kidney damage and neurological damage. Standard treatment for this particular disease is a high-dose chemotherapy followed by a stem cell transplant. So how do you get from normal to multiple myeloma, myeloma-defining events, and beyond? Well, the disease process is under the umbrella that we call plasma cell dyscrasia. On this diagram, from left to right, we're going from normal and healthy to further severity of disease. So increased disease severity, increased mutational burden in the plasma cells, and increased genetic instability in those cells as well. In normal cases, there is some serum protein and a low number, but not zero, of plasma cells within the bone marrow, and of course, no myeloma-defining events. Plasma cell dyscrasia becomes to be detectable when it presents as a monoclonal gammopathy of undetermined significance. And what's happening is that there is a single antibody which is being secreted in the bloodstream, which is increasing the amount of serum protein. The amount of plasma cells in the bone marrow is also increased at this point, but it's less than 10%, and no myeloma-defining event has occurred. The next step in the process is smoldering multiple myeloma. We see a further increase in serum protein as the tumor is growing and secreting more protein and more antibody into the blood. Plasma cell percentage in the bone marrow can now be as high as 60% of the cells being these plasma cells, but no myeloma-defining event has occurred. So no anemia, no bone breaks, nothing like that. Full-blown multiple myeloma or active multiple myeloma meets the same criteria, only there has been a myeloma-defining event. From here, the disease can progress into what we call extramedullary disease, or EMD, and this is where the multiple myeloma moves back out of the bone marrow into forming tumors on secondary organs, or even plasma cell leukemia, in which case more than 20% of the white blood cells in the patient are multiple myeloma cells. So how is this monitored and diagnosed in our patients? Well, the diagnostic pathway begins with a standard blood test and a host of cellular assays and molecular assays on bone marrow biopsy. Treatment selection is then made, and then testing is repeated during monitoring and remission if treatment is successful in these patients. And what I'd like to highlight here is that at diagnosis, treatment monitoring, and in remission, we're talking about a bone marrow biopsy. This is an invasive, painful procedure, and it is injurious to the bone, which is already dysregulated in multiple myeloma patients. So this underscores that we had a need here for a non-invasive modality for assaying what's happening in this disease in these patients. So why the need for monitoring in remission and in treatment? Well, the cycle of multiple myeloma is presented here. On the x-axis, we have time. On the y-axis, we have M protein or myeloma protein. This would be the antibody secreted by this disease as a sort of placeholder for disease severity. When the patient presents and is diagnosed, therapy is applied, and it reduces active myeloma into a remission. Now, unfortunately, virtually all multiple myeloma patients will relapse. So you get a return to disease, therapy is applied, to bring it back into remission. This happens again and again and again until eventually the relapse in the patient is refractory to treatment and a massive paradigm shift in treatment needs to be made. So what's happening during these plateau remissions? Why is this so difficult, and why are we not able to cure these patients? Well, one of the big contributors to this is what we call minimal residual disease, or MRD. Presented here are two graphs comparing patients above and below the cutoff for MRD, or minimal residual disease, which is fewer than 1 in 1 times 10 to the 5th cells of plasma cells in the bone marrow. And what we can see on the graph on the left, we have time on our x-axis and progression-free survival on our y-axis, is that those patients which are below that cutoff have an improved progression-free survival. The same is true when looking at overall survival. Similar graph, time on the y-axis. On the x-axis, we have cumulative survival in percentage, that MRD negative patients have an improved overall survival compared to those that have MRD or plasma cells above that cutoff. So in monitoring MRD, today, this requires multiple bone marrow biopsies. And again, this is deeply problematic as multiple myeloma patients already have dysregulated bone health. In the future, the EMA draft guidelines suggest that we need to find a way to monitor MRD using peripheral blood and not bone marrow biopsy. And so in the future, the ideal would be a serial blood draw test to monitor MRD and replace this bone marrow biopsy as the standard treatment modality. The challenges in multiple myeloma are the same as those for MRD. Virtually all of our patients are going to relapse, and so this means that this requires monitoring of these patients. And the goal of the treatment here is remission, not a cure, because of that MRD problem. Monitoring of multiple myeloma is extremely invasive because it requires this repeated bone marrow biopsy. This again injures the bone in patients which already have disrupted bone health. And one other huge problem with this is that testing is initially done usually at the onset of symptoms, at which point it's late in the disease process and may be too late for proper treatment intervention. So there is an urgent need in this field for a non-invasive way to look at plasma cells. And so what we've come up with is a way to do that, to measure circulating multiple myeloma cells, or CMMCs, using a technology called CellSearch. So the CellSearch CMMC assay involves counting multiple myeloma cells in a single blood draw to monitor disease progression in a clinical research setting. So this is an RUO kit. So let's talk a little bit about CellSearch and how it works and why it works. Well, CellSearch is the gold standard for monitoring circulating tumor cells. It's the only FDA-cleared and CE-IVD-marked blood test for enumeration of circulating tumor cells, with over 150 patents and 600 patent applications, and it's backed by over 600 publications in peer-reviewed journals. The IVD test is the first and only actionable test for CTC counting in cancer patients with metastatic breast, prostate, and colorectal cancer. So the CMMC test is built on this backbone to look at multiple myeloma cells instead of metastatic breast, prostate, or colorectal cancer. So built on the CellSearch platform, we developed an assay to count circulating multiple myeloma cells from whole blood. The blood is collected in the cell rescue preservative tube, which I'll talk about on the next slide. And this, combined with the staining reagents, is combined into the autoprep instrument, at which point the cells are isolated, fixed, permeabilized, and stained for fluorescent enumeration downstream. The autoprep adds these cells to the Magnest cartridge, which actually organizes all the cells into a single focal plane so that it can be automatically imaged by the CellTrax Analyzer 2. You take the Magnest, add it to the analyzer, image it, and then you can do your enumeration to count these cells of interest, which in this case are circulating multiple myeloma cells. So I mentioned the cell rescue tube. What is this and why is it so important? So in order to preserve sample integrity and to maintain surface marker expression of the CD138, specifically for the CMMC assay, we developed a novel sample collection tube to meet these needs of researchers interested in interrogating CMMCs. This is built on the already excellent CellSave tube, and both CellSave and CellRescue offer 96 hours of sample stability post-collection at room temperature, meaning that you can collect your samples and they'll be good to go for up to 4 days. So, combined with Cell Rescue, how then does CellSearch capture and stain our cells of interest, in this case our circulating multiple myeloma cells? Well, this is done with antibodies bound to ferrofluid. In this case, the ferrofluid is bound to an antibody against CD138, which is a surface protein on plasma cells, especially for multiple myeloma. Included in the staining kit is a secondary positive marker for CD38. This will specifically highlight multiple myeloma cells and differentiate them from normal plasma cells in the assay. Also included in the staining reagents for this kit are a cocktail of antibodies which stain against CD45 and CD19 in order to exclude any normal white blood cells. CMMC capture and detection is done using 4 mL of peripheral blood, and we define a CMMC as a plasma cell which is CD138 positive, meaning it's bound and isolated by our CD138 ferrofluid, that stains with our CD38 marker, that is DAPI positive, but is negative for CD19 and CD45. And one thing that's important about the CellSearch system is that it's a 4-channel fluorescent system, and there are only 3 in use right here. This allows us to add, or it adds the capacity for, a user-defined marker. So if you have other markers of interest to you in your studies, there is an available channel for you to add those in order to obtain more information beyond enumeration and identification of CMMCs in your studies. So now that we have the ability to isolate and enumerate CMMCs, how do we use these as a biomarker, and what have we learned? So when we looked at CMMC count in multiple myeloma patients, here's what we found. Presented here are some data on CMMC count in patients. In the red, we have data of newly diagnosed patients, baseline. In the green, we have remission patients, so patients after treatment. And then in the blue, those are the counts for patients that have relapsed. We have sort of 3 blocks of graphs here, and those are ISS stage 1, 2, and 3 from left to right. On the y-axis is just number of CMMCs in a 4 mL draw, and across the x, that just is a further description of which subplot you're looking at. Now what you can see, if you look at the data presented in red, is that overwhelmingly newly diagnosed cases in samples drawn at baseline showed CMMCs. And in some cases, there were thousands and tens of thousands of CMMCs in the patient sample. At remission, after treatment, we see a decrease in CMMCs, like we would expect. And upon relapse, we see a return to higher levels of CMMCs. This demonstrates that there is potential here to utilize CMMCs to monitor disease progression. Well, how does CMMC count relate to the outcome for these patients? Can it be used as predictive? And in this case, we took the same patient cohort, analyzed them, and found that we were able to striate CMMC count and outcome with a cutoff. So this is a Kaplan-Meier plot. Progression-free survival in percentage of patients enrolled in the study is on our y-axis, and time in days is on our x-axis. We can see 2 pretty distinct trends here. In black are patients that had fewer than 100 CMMCs during remission, and in red are patients that had more than 100 CMMCs at remission. And we see a pretty distinct correlation to higher CMMCs above our experimental research cutoff here than those that were below it. Lower CMMCs during remission is a better progression-free survival. Okay, so we know about remission. What about in other stages of plasma cell dyscrasia? How does CMMC correlate to things like MGUS or smoldering multiple myeloma? The data here presented on the right side is CMMC counts compared to healthy donors on the left, so that's healthy, normal blood, and that's dozens of patients. In the middle in blue, we have smoldering multiple myeloma and MGUS patients, and then on the right, this is compared to full-blown active multiple myeloma. What we can see from the data here is that compared to healthy donors, smoldering multiple myeloma and MGUS patients are sort of between healthy donors and full-blown multiple myeloma patients. So this suggests that we can think about CMMC count and even the precursor conditions to multiple myeloma. The current strategies in monitoring this in patients, if you develop monoclonal gammopathy, is if you have no other symptoms, sort of watch and wait. If it's suspected for some other clinical presentation that there is smoldering multiple myeloma or multiple myeloma, then you progress to imaging and biopsy. With the addition of information about CMMCs from our kit, if you have a gammopathy with a low CMMC count, the watchful waiting paradigm represents a fairly low-risk strategy. So that is probably something you could reasonably undertake. But this suggests that if you have a gammopathy with a high count, that watchful waiting here puts the patient at risk. Now again, this is all in an RUO setting, but it's pretty suggestive that you can get really meaningful information about even precursor states of multiple myeloma with the CMMC kit. So, how do you bring the CMMC test into your research studies? Well, you do that by partnering with us. The CMMC assay is available only from MSB Global Lab Services. MSB Global Lab Services offers the CMMC test, and this can be run at our facilities either in the United States at Huntington Valley in Pennsylvania or at our facility in Bologna, Italy. This is especially useful since our CellSave and the CMMC test's cell rescue tubes ensure a 96-hour sample stability. This means that if your site is within a 4-day shipping window of either of our facilities, we can take your samples on, run them, and analyze them. Our Global Lab Services team has extensive experience in clinical studies, clinical services, and in pharma and biotech. We've been doing this for more than 13 years, serving the global market. And we have a proven, demonstrable track record for our quality audits, and it's powered by our CellSearch tech and our DEPArray. So we are always able to help you run the samples and run the studies that are needed for your studies and in your clinical research. Submitting samples to our Global Lab Services looks a little bit like this. First step is to collect your blood in one of our 96-hour window tubes, either CellSearch or Cell Rescue for the CMMC test. This is then shipped to us. We will then take the sample, test it, and analyze it, and provide the data to you. Time from sample receipt to turnaround of data is about 2 days. There's an option here as well to take the sample, freeze it down, and to thaw it for future analysis, be those molecular or other analyses of your desire that we can offer, and for us to provide those results to you as well. Our CMMC test demonstrates tremendous potential to meet this unmet need as a liquid biopsy biomarker for multiple myeloma, and we at MSB are positioned like no other company can be to be the global partner for you and your research lab to study CMMCs in your studies.